you were you presented as a student, I think, right, at Columns?
Yeah, I think it was honestly only like 3 years ago. So time flies. But
He's an up and comer. So Byron, he's now an aquatic scientist over at SePRO. He's working out of Sacramento in Northern California. When did you move up there?
just recently, over in June.
Cool. And so Byron's got a pretty long list of accolades. He's got a PhD. He got it from UC Merced, working under Marc Beutel. He was studying redox and oxygen control on water quality. He has a BS in chemistry, an MBA, and a master's degree in environmental engineering. So we've got a very qualified speaker with us today with a lot of knowledge. And is there anything else you'd like to introduce about yourself before we get started?
Not much. I would say my passion is really for sediment biogeochemistry. So I will say that Eli is the expert in some of the topics that we'll be discussing today. So I'm going to pull him in, and we'd love to hear from everyone as far as like your experiences and your take, and just your opinions on lake management in general. So, if you have any questions or some insight from previous experience, feel free to type that in the chat box.
Yeah, I'll be watching the chat, and Byron, I'll kind of interject if there's any questions that come up that are relevant. But everyone else, I'll be watching the chat too. So throw some questions in there, and when we get to stopping points, I'll make sure that we get those asked publicly. So we'll look forward to hopefully a good interactive talk about algae. And take it away, Byron.
One more quick announcement first. This is Mark again. I just wanted to let everyone know that this meeting is being recorded and that you'll be able to access the recordings at columns2020.org.
Right. And one other last announcement is please consider donating to the Student Scholarship Fund. We have, as you see in the background, thank you to all our sponsors who have contributed. And we had a couple other members and just viewers donate a couple bucks. Anything helps. 25 bucks, 10 bucks. Got a lot of folks put a lot of effort into putting this on, and all we're asking is donations for students. So feel free to click donate on the website. All right, take it away.
Yep. And I will add, we are a nonprofit. So if you guys need to reduce your tax liability and put your money towards something, beneficial, improving lake management and lake water quality around California would be a great way, and to support some of these up and coming lake managers, and help us give more student scholarships in the future. And thanks again to our sponsors. So everyone listed on the screen, hopefully you guys can see it, but they're really helping us to continue as an organization. So a lot of really great lake management groups, some utilities like Valley Water, a lot of companies that work in water quality analysis and water probes, some consulting firms like McCord Environmental, and some interesting newer technology like floating islands. So just wanted to give a shout out to all of these people that supporting our sponsors indirectly supports us, because they can say, hey, we really benefited from supporting an organization like Columns. So we'd like to get their name out there and go check out some of their products and some of the services that they provide. So my talk today will be on eutrophication solutions, and really how we can learn about algae management from the past, and some of the things that we've learned throughout the past century of doing algae management, and maybe take advantage of some of these novel insights. But first, we're just going to start at the very basics, where we're just going to ask, what is eutrophication? So trophic status, where it's either oligotrophic, mesotrophic, eutrophic, or even hypereutrophic, is really determined by the amount of organic matter in each water body. So oligotrophic is just very crystal clear blue water with very low organic matter in the sediment. If you could grab some sediment from a place like, say, Lake Tahoe is a perfect example of an oligotrophic lake, you're going to find a lot of sand, silt, and clay, and not very much organic matter. So on the opposite spectrum, a eutrophic or even a hypereutrophic lake is going to have that lime green Kool-Aid type of water. You're probably going to have some algae scums. organic matter throughout the water column. And if you grab some of that sediment, you're going to see a lot of more black stuff, a lot of muck, some partially degraded organic matter. And mesotrophic is kind of that intermediate stage. So there's natural lakes of all three of these types, but in general, most lakes are oligotrophic or mesotrophic naturally before humans started messing around. And you know, if it's not a natural system, so something like a reservoir, these don't get built so that they can be eutrophic or hypereutrophic. They're originally designed to hold water for a purpose, and usually it involves recreation or drinking water. So oligotrophic is by far the better option for these. But when you have a lot of human activity, we're really just having the input of a lot of nutrients into these systems, and these nutrients are going to fuel the growth of algae at the surface. And once you start growing a lot of algae, you can kind of think of a lake as a closed system, and it's very difficult to get rid of that organic matter. So too much algae is going to die and decompose and end up in the sediment and cause all of these problems. So really, we want enough algae to fuel the food web, but not enough so that the food web can't handle it. So there's a decent amount of side effects for eutrophication. I mean, the obvious one is it's ugly. Like, no one thinks of a lake with this patch of green stuff all over the surface is just beautiful and awesome, and that's not the best place to go swimming. So, you know, you have a lot of recreational value reduction in these. And you have a lot of toxins that are associated. So the more algae you grow, generally, the more cyanobacteria you're going to have. And these HABs, or harmful algal blooms, are associated with just the high abundance of cyanobacteria, when a lot of these are making toxins. So the more research we do into these HABs and this toxin production, the more we're learning about the side effects and how managing these HABs is very, very important. So, you know, like Keith from the Water Boards, he's doing a lot of great research on that. And, you know, we should really support the further research into HABs and how to mitigate these and how to understand some of the risks associated. But these toxins can accumulate in fish and shellfish. I mean, they can get to concentrations where they're actually lethal to dogs that are going to go in and drink this water, or even wildlife like deer. And it's a high risk for children, for pregnant women, and even just for people in general. And clearly it's not good for drinking water, because, you know, it's so much easier to purify some water that's already, you know, crystal clear blue, rather than stuff with organic matter all over it, not to mention all of the stuff that gets released from the sediment during eutrophication. So I think that it's very highly just not as well understood how big the relationship is between the food web and eutrophication, because we think of it as, okay, everyone knows like a lot of nutrients are going to grow a lot of algae, which is the phytoplankton on this little graph. But from there, you know, if you have a very balanced food web, then you can stimulate the uptake of this algae into fish. So essentially, you're putting nutrients into the fish biomass. And then if fishers are you know, catching those fish and taking them out, then that's a benefit of putting nutrients into the system. Whereas if you're just growing a lot of algae and the food web really can't handle it and eat everything, that's when you get into this vicious positive feedback loop of, you know, having that algae degrade and release more nutrients. So it's kind of complicated, but I'd like to just go through the aquatic food web really quickly. So at the base, you have the nutrients, which are growing the algae, and the zooplankton are going to eat the algae, and the prey fish eat the zooplankton, and then the predatory fish eat the prey fish. So it's a very simplistic model, but I think it demonstrates most of the points rather than getting into something that's super complex. So starting off with the nutrients, that's really going to be soluble nitrogen and soluble phosphorus. So soluble nitrogen, there's going to be two major forms of that, and it would be nitrate, which is the oxidized form, and ammonia, which is the reduced form. So whenever you have oxygen around, you're generally going to have a pretty quick oxidation of ammonia into nitrate. So in surface waters, nitrate's a little more common. And algae do have preferences for certain types. So some algae can use both. And in those instances, ammonium is going to be more efficient. It's easier to cross the cell membrane. It's easier to incorporate into proteins. But the advantage of being able to use both is that you don't have to rely purely on ammonium. And so soluble phosphorus is really only in the form of phosphate. There's not all that many algae that can utilize organic forms of phosphorus. So that makes it a little bit easier. Now, if we move up the food web into the beneficial algae, the two that I'd like to talk about are green algae and diatoms. So there's a lot more types of algae, but these are, some of the staples and some of the most popular. They both get eaten by zooplankton, so they can both transfer those nutrients in the form of their biomass into zooplankton, which can eventually become fish biomass. And they both don't release toxins into the water column, so at least in freshwater systems. So it's beneficial in general to have, moderate amounts of these algae. The green algae have a more classic appearance, whereas the golden algae the diatoms are gonna have more of a golden or a brown colored appearance. And the diatoms are really cool because they have these silica cell walls. So they polymerize silicon dioxide, which is, just like sand, and they use this to form their cell wall. And that's actually more energetically efficient than using like organic matter, because green algae and other algae, they have to use organic matter to make a cell wall. And that requires photosynthesis and using some of that organic matter that they're fixing from carbon dioxide. So if you have a lot of silica in your water column, it could stimulate some diatoms, and they can be very efficient if they have the right conditions, because they also are gonna need nitrate. They don't grow very well, if at all, with ammonium, whereas green algae can generally use both forms, and then they're gonna obviously prefer ammonium since it's easier to uptake and to incorporate. So now, instead of moving up the food web, and here's some pictures. So, green algae, a very popular form of this would be Pathophora, and it's called like cotton algae because it has like a cotton-like appearance. So these can be good. I would say the amount shown in these pictures is not good because that's too much for zooplankton to eat, and it's eventually going to die and sink to the sediment and decompose and cause some problems that we can talk about a little bit later. Whereas diatoms are on the right-hand side and in the middle, and you can see that like golden, kind of brownish color to the water. So that's most likely a diatom bloom. They have these really, really cool, intricate cell structures. So I think they're really amazing, and there's so many different species of diatoms. Their use of silicon dioxide to form these cell walls is super interesting, and it allows them to do these things. So if you're interested in that stuff, I would go, just Google some different diatom species. So rather than going up the food web, we're going to go to the side, and we're going to talk about blue-green algae, which I would consider as disconnected from the food web, because in general, zooplankton don't really like to eat them because they're producing these toxins. So these toxins are an adaptive mechanism to get their predators to not want to eat them. It's just kind of a coincidence that humans also find these as toxins as well. So it's really done so that they don't have any predators. And the problem with them not having any predators is that they're going to be able to survive and then live out their life cycle, and then they'll die and sink and decay and just release nutrients back into the water column rather than sending it up that food web. And so they have a lot of cool advantages as well, which makes it more difficult to kind of control them. But they like to fix nitrogen gas. Not all of them can do this, but there's some very popular blue-green algae that are very good at fixing nitrogen gas and living in very low nitrogen conditions. So this is an advantage if you have ample phosphorus in the water column, but you don't have very much soluble nitrogen. They can also, a decent amount of them can regulate their buoyancy with gas vacuoles. And so when they do this, they can either sink or they can float on top of other buoyant things like green algae or diatoms. So when they sink, they can go access the bottom water nutrients, and then they can really become very buoyant and then get on top of the other algae and outcompete them for the light. But they're also adapted to low light, which is very cool and a good strategy because they can start before the other algae can start. When you have that transition from winter to spring in those pretty low light conditions, if you have enough nutrients, you can stimulate a cyanobacteria bloom rather than stimulating those nutrients to go into green algae or diatoms. So here's some pictures of cyanobacteria. Usually they look like kind of like oil slicks with like, it's green. And a lot of them are, they don't form these big filamentous colonies. So like stuff like Anamina or Microcystis, you could put your finger in there and it might attach to your finger, but it's not going to form these big clumps. An exception to that would be Lingbia. So Lingbia is over on the left-hand side, and it's like, it's these really long strands and like hair-like and silk-like kind of. And when you get Lingbia in a water body, it's very difficult to get rid of. It doesn't degrade very well, so it'll just accumulate on the bottom of the sediment. So here's just a little compare and contrast of all the diatoms, the green algae and the blue-green algae, some of their unique features and some of their use of nitrogen. So starting with the diatoms, the most favorable conditions to grow some diatoms would be a lot of silica in your water and then a lot of nitrate. If you have those two conditions, you're probably going to be growing a lot of diatoms. For those reasons, they're popular in like saltwater fish tanks. Now, if you have lower amounts of silica, you're giving the blue-green algae and the green algae more of an advantage over the diatoms. And so the green algae are going to have an advantage when there's a lot more either ammonia or nitrate. So just a lot more soluble nitrogen for them. And that takes away the advantage of nitrogen fixation by the blue-green algae. The blue-green algae are going to be favored when you have a very high phosphorus levels compared to your nitrogen levels, because that gives nitrogen fixation a very clear competitive advantage. And they can deal without having any nitrogen in the water column. They're all going to uptake that phosphorus and they're going to fix nitrogen from the atmosphere. A lot of times too, they like when there's nutrients being released from the sediment in a deeper water system, because they can take advantage of that adaptation to regulate their buoyancy. So moving up the food web, we go into what I would consider the key intermediates. So the zooplankton, having a really strong, robust, diverse zooplankton community is very important to keep that algae in check. And so if you're not growing too much blue-green algae, maybe by monitoring or regulating your nitrogen to phosphorus ratios, which we'll talk about a little later, and you have a very big zooplankton community, which diversity and just really abundant would help a lot, because they can eat a lot more types of algae. That's going to help that algae organic matter not to accumulate to the point where it's going to sink and end up in the sediment. And instead, it's going to send those nutrients through the food web up into the fish. So next, moving to the top of the food chain, and I'll group these together, even though they have very different effects, but the fish kind of regulate this entire system. And so what you don't want is too many prey fish. If you have just a huge community of small fish, then they're going to decimate that zooplankton community. And keeping this zooplankton community, to be very, very abundant and to be diverse and to be spread out throughout your entire aquatic environment is really important to control that algae, as we talked about. So predatory fish, a lot of times stocking this can be very advantageous, because they're going to keep these prey fish in check and allow this zooplankton community to really blossom. And the predatory fish, like the largemouth bass, they don't care for zooplankton. They need some real meat. So sometimes if you're low on your high trophic level fish, it's a good idea to stock some of these to keep those prey fish in check and to moisture your zooplankton community. So just in summary about eutrophication and the aquatic food web, I think of beneficial algae being stimulated over blue-green algae. That's just improved aquatic ecosystem function, and that's really strengthening your food web and the ability of you to promote the transfer of nutrients into fish. And that's really the goal. That can't be, for a lot of eutrophic and hypereutrophic systems, that's not the end-all solution to everything, but that really helps to regulate and to be able to buffer the input of nutrients every once in a while. And with less blue-green algae, you're going to have a lot less toxins, and you're going to have that ability to transfer nutrients into the fish. So you might be asking, what causes eutrophication? Well, I think it's moderately simple. There's three main factors. Light is going to be number one, and that's the reason why you never really see algae blooms in the wintertime. There's just not enough light to drive that photosynthesis that it needs for algae to grow and to reproduce. So if you have enough light during the springtime, and remember the cyanobacteria can deal with slightly less light, so they might have that competitive advantage to come out first when the season's changing from winter to spring. Once that's happening and they have enough light to drive that photosynthetic need, now they're going to need nutrients. And so cyanobacteria might be able to do without nitrogen, but they can't do without phosphorus. They can't fix their phosphorus. There's no gas form of phosphorus that's significant. So really, by regulating the amount of nutrients, you can have all the light you want and you won't actually grow any algae, which would be a very good idea too. And that's why it's important a lot of times to regulate phosphorus a little more so than nitrogen. So finally, the last one that's important, it's certainly not as important as number 1 or number 2, is temperature. And it's related to the light intensity, but that depends on elevation. So at some higher elevations, you're going to have colder temperatures, even if you have more light penetration. But temperature is really just related to your metabolic rate. So if you're really warm, and algae, are just like humans or just like other animals, the warmer you are, the more your metabolism is going to be firing, and you're doing a lot of metabolic processes. Your cells are growing and dividing and reproducing. When you're colder, you're more in a dormant or a hibernating state. So next we might ask, well, what? I'm sorry, I'm going the wrong direction. So yeah, talking about nutrients, phosphorus is just a little bit more important, and so there's this famous ratio of the Redfield ratio, where Redfield went around marine environments and sampled a lot of different algae and found that there was this tight ratio of 106 carbon atoms to 16 nitrogen atoms to 1 phosphorus atom. And so there's some newer research that is saying that this isn't right, but it's in the general ballpark of what we would expect in most life forms. And so if we include hydrogen and oxygen in this, that comes to about 500 parts total for 1 part of phosphorus. So that means that 1 pound of phosphorus could, in theory, grow 500 pounds of algae. And especially if it's cyanobacteria, if you're going into a lake that doesn't have any nitrogen and you just put 1 pound of phosphorus, it's pretty much all going to be cyanobacteria, because they're going to fix their nitrogen from the atmosphere. And you're going to get 500 pounds for 1 pound of phosphorus. So phosphorus control is pretty important. And nitrogen is pretty prevalent in the environment anyways, because we have a lot of NOx emissions from the burning of fossil fuels and from gas and all of this stuff. So when these NOx emissions get into the atmosphere and get into the clouds, they come down as a lot of times nitrate. So your rainwater might actually have some input of soluble nitrogen, even though it's not going to have any soluble phosphorus in it. And you also have nitrogen fixation by blue-green algae or cyanobacteria, and then a lot of bacteria that are going to live in these more like littoral zones in symbiotic relationships with some of the wetland plants. So phosphorus limitation is going to be a lot more common, and it's just a fact that there's no real phosphorus cycling. There's no gas phase of phosphorus. So this brings me to the N/P ratio, where remember, our 16:1 is our standard Redfield ratio in terms of nitrogen to phosphorus. So a low N/P of less than 10:1 is going to favor blue-green algae, because it's going to make nitrogen fixation a really good adaptive characteristic. And it's going to say, hey, you can get by without having any nitrogen in the water column. Just take that from the atmosphere. So they're going to be able to outcompete the other types of algae. Now, the high N/P ratio, something like more than a 30:1, is going to favor stuff like green algae and diatoms. And that's because now nitrogen fixation isn't such a great characteristic, and it's not going to actually be used because it's pretty energetically intense to fix nitrogen from the atmosphere. So in these situations, green algae and diatoms are actually favored. So by having a really high N/P, we can allow the trophic transfer of these nutrients into the fish as these beneficial bacteria get eaten. So really a low N/P and you're looking for some algae scums and some HABs, and then a higher N/P and you're looking for some big bass. Also, with eutrophication, there's this redox feedback loop, which can be really, really vicious in a lot of these hypereutrophic systems. But the general concept is that you're growing too much algae, and then that organic matter accumulates in the sediment after the algae dies, and it's too much for the zooplankton to eat, or it's a lot of cyanobacteria. Now, when this organic matter goes to the sediment, the bacteria in the sediment are going to oxidize that organic matter. And the way that they oxidize that organic matter depends on what oxidants are available. So if there's any oxygen around, that's going to be the most energetically efficient and the most favorable mechanism to burn through that organic matter, and aerobic respiration is going to be favored. But these aerobes, or the bacteria that use oxygen to oxidize organic matter, they are going to rapidly use up all of the oxygen, and sometimes this can be the whole sediment water interface and the bottom water. So once the system runs out of oxygen, now it gets pushed down into denitrification. And denitrification is a great redox stage. And you might hear about, oxygenation or nitrate addition, because it poises that redox above the other stages in the redox ladder, because really the only byproduct of denitrification is nitrogen gas. But once you get below denitrification and you use up all of the nitrate, which algae are trying to use that anyways, now you get into manganese reduction, and that results in dissolved manganese being released, which is really a problem for water treatment. And then below that, you get into iron reduction. And so solid iron oxides, they're great at binding phosphorus. But once you reduce them, now they're going to release that phosphorus and they're going to release dissolved iron. And cyanobacteria also have a high need for iron, so that will continue to stimulate them. And then below that, you get down into sulfate reduction. And so these sulfate reducers, they produce sulfide, which is a gas and it's toxic. Not only that, but they also like to create methyl mercury. So there's a great talk on Thursday about using oxygenation to potentially reduce the mercury levels in fish by keeping that redox above the level of sulfate reduction. So really encourage you guys to check that out. It's going to be by Marc Beutel, and a lot smarter than me, so I can't get into all those details right now. But in general, just going down this redox ladder is going to be bad, because we're going to release things like nutrients, which are going to grow more algae. And then when that algae grows and dies and decays and goes back to the sediment water interface, you're stimulating the reduction in redox again. So here's just a little animation of all of this happening. So as the algae gets eaten, some of it decomposes and goes to the bottom of the sediment where the bacteria start eating it. And that drops the redox and it turns iron phosphate into the soluble phosphates, grows more algae, and now you see sulfide is being emitted to the surface water as well. And we're going all the way down into the methanogenic conditions and some of the potential water quality contamination that comes with that. So now we're going to get into, how can we fight eutrophication? What are some short-term solutions for eutrophication? I'm dealing with some algae blooms and I want to get rid of them. I want to have a nice lake, in the next couple weeks or so. Well, you might not have a couple weeks with watershed management, but watershed management is the key to fighting eutrophication. If you continue to allow nutrient input into your water body, you're just asking for problems, regardless of what your other strategies are. So we really just need to manage our point sources, which are just identified as major sources. And there's a TMDL, or total maximum daily load, that is applied to a lot of these. And so this is calculated at how much can the water body actually use and buffer. After that point, it usually goes into growing too much algae and having eutrophication issues. We also need to worry about non-point sources, which are a lot tougher because they're harder to identify. But things like wetlands can be a great barrier, because wetlands can really do a lot of nitrogen cycling, and you can stop a lot of the phosphorus if it's in the form of particulate phosphorus and bound to sediments and to organic matter. And then there are also some nutrient barriers which can bind phosphorus or nitrogen compounds. And so if you can identify which streams or tributaries are bringing in your nutrients, these barriers could be a great mechanism. And best management practices is like managing stormwater. And so that's really important, because a lot of people just use too much fertilizer. And so when it rains, they'll pump all of that into the storm drains. And then if that storm drain connects to a water body, you're looking for eutrophication down the road. There's also some food web management. I would say this is the stuff that it's helpful, but it's not the end all solution. So, you know, biomanipulation, like we talked about, and you can stock some big predatory fish to keep your small fish populations lower and to enhance that zooplankton community. You can have carp harvesting. So carp like to swim around in the bottom and they stir up a lot of sediment. And this stirring the sediment releases a lot of nutrients into the water column. So taking out carp for a lot of systems has a lot of benefits, and you don't have as much nutrients being released into the water column. Some floating wetlands. I think this is a really cool concept. They're not as great for something like phosphorus removal, but they're pretty cool for nitrogen cycling, because you're basically having a wetland that's floating. And the other advantage of these is that in these root zones, you're giving a sanctuary to a lot of zooplankton, because they like the shade, where a lot of fish are sight hunters and they can't actually see the zooplankton. And then it's also going to have a cooler water. So in a lake or a water body where you have, you know, like anoxic bottom water with very low oxygen levels, but then you have hot surface water, this could kind of serve as that refuge for them. And, you know, it's one of our sponsors is Floating Islands West, and they have some cool designs. I think the other cool thing about floating wetlands is they're very aesthetic and they're not super expensive. They might not have the largest impact. I don't think that these could necessarily just save a eutrophic water body and make it oligotrophic, but it's on the right steps, and it's, you know, one of these natural remedies. Another one would be vigorous epilimonetic mixing, so manipulating this food web, and you're discouraging blue-green algae growth by vigorously mixing the surface water and the bottom water. So sometimes it's with a pump, sometimes it's just through aeration. But by doing this, you're making these blue-green algae that like to regulate their buoyancy, you're kind of confusing them. And when they start doing that, they get caught up in this cycle and they get mixed around, and it makes it really hard for them to photosynthesize. Whereas the green algae that just like to kind of hang out at the surface water, they're favored in this process. And so the gold standard of the short-term solutions for eutrophication is really the algicides. And that's copper sulfate, chelated copper, and peroxide based. And this is like 99% of the algicides that get used. So here's just a little bit of details about all of them. You know, I've tried to make a quick little pros and cons and best fit list. Hopefully it's not too much information. But copper sulfate was like the original. They found that, you know, copper really ripped through these cell walls of it's toxic to a lot of plants and algae. So for irrigation, you know, it's generally like frowned upon to have very much copper, and usually these water bodies are regulated. But copper sulfate, it really gets quickly removed by reacting with the carbonates and the hydroxides in the system, and it will precipitate out. And then when it interacts with organic matter, it's usually going to be reduced into a less soluble form of copper. So copper sulfates, it's quick, it's fast acting, and it's cheap. And so, you know, that could be a good thing or it could be a bad thing depending on the water body. But it's not very effective in the hard or high alkalinity waters. And then it can be more toxic in the really soft waters and have a higher non-target toxicity to things like fish. So it's the best fit for really like a low budget and a less sensitive environment. You know, if it's just a pond and you don't really have any fish or care about the fish, and you just want to make it look clean, you know, copper sulfate, and you don't have a huge budget, copper sulfate might be your best bet. The chelated coppers, it's kind of trying to take advantage of the weak points of the copper sulfate. So you're going to need a lower dose of copper, and it's going to stick around a lot longer. So these copper ion has organic ligands that bind it and keep it from reacting with things like carbonates and hydroxides and precipitating out really quick. So you'll have it hanging out in the water column a lot longer. It can diffuse around, it can be diluted. And so it's going to work a lot better in the hard and the high alkalinity water. And it's also going to have a lot lower non-target toxicity. Eli can talk just a little bit about this briefly. So it's going to, since it hangs out in the water column, then it's going to have a higher persistence. So it'll be harder to meet your copper compliance if you do for, you know, irrigation or for discharge. And it's going to have less of an instant impact compared to copper sulfate. So it's like a longer term, maybe a week's worth of algicidal activity versus copper sulfate, which is, you know, going to be pretty immediate. So it's going to be the best fit in like a harder, high alkalinity water where copper sulfate might not work. probably no strict regulations on copper and dissolved copper in the water column. And then where you're going to need a little bit longer term control. Now, peroxide based is, I think it's really interesting, and it's oftentimes a really great alternative when you have regulations that don't allow for copper. And it's really cool too because it's toxic, more toxic to cyanobacteria than it is to green algae and diatoms. The cyanobacteria are a little bit more simple as prokaryotes. And so they're actually affected a lot more by the oxygen radical that is created when peroxide reacts in water. So it's really fast acting though, that this peroxide really only persists for like minutes at the most. So that makes it tough because it's not going to be able to dilute and diffuse around your water body. You really need to directly hit the algae where it is. But there's no irrigation restrictions, and there's basically no non-target toxicity if you use it as the label directs. But usually pound for pound algae control, it's going to be a lot more expensive than your coppers. So it's going to be the best fit for like if you do have a higher budget and a more sensitive environment. And even, you know, if you're highly regulated and you know, if you want to keep those green algae and those diatoms and try to work with your food web to uptake those nutrients and put it into fish. So now I'll let Eli talk about some more complicated aspects. He's really the algicide expert.
I guess so. Byron, thank you so much so far. It's been a lot of help. I think it's a very comprehensive view, and I think it's definitely the approach people need to take. Can you guys all hear me? Can you hear me okay?
Yep, we can hear you.
So my license I have is called the Pest Control Advisor's License. And often when you do want to use a chemical approach, a chemical herbicide or algicide, it's good to work with a pest control advisor. In California, you're really not supposed to make recommendations on chemical usage in a lot of situations unless you have the license. But I will say the main focus that I always take is this integrated approach. So let's look at the system-wide approach. Let's look at the biology of the pest and try to find ways to prevent and reduce the growth as much as possible. Hang on one second. So with regards to coppers, you brought up some good points. Would you actually go back one slide real quick? So we're really talking about two types of coppers. We've also talked about peroxides. There are some other algicides as well available out there. There's endothol-based algicides, but often this is kind of the first step is, do we use a copper sulfate or a chelated copper when we're going to address some sort of an algae issue if we are going the chemical route? And I'll just bring a couple of things as Byron had mentioned. One of the big benefits is reduced irrigation restrictions. So this is often a good solution to use in irrigation ponds and irrigation ditches, canals. But he brought up an interesting point here, which was talking about hard water and alkalinity. And so if you skip forward again now to the next slide, when we start thinking about using these different formulations in your lakes and ponds, we need to really be focused on what is the water quality conditions, specifically with hardness and alkalinity. As he mentioned, the copper ions will bind with other minerals in the water and precipitate out. And if we have very little calcium, magnesium, iron, and other minerals in the water, the amount of copper that you apply can quickly become toxic to fish and other non-target organisms. It is a biocide. So copper sulfate particularly, you know, we've been using it for centuries as a fungicide, as an algicide, to control mildew, molds in crops, as well as in water. So it's kind of, it can be pretty indiscriminate. And so I think Byron made a really good point talking about the zooplankton, and there is the potential to really harm that community when using coppers, especially if you're overusing coppers. And I think that the main takeaway here is that overuse is highly dependent on the quality of the water. What is the environmental impact on the chemical you're using? I think a lot of times we often think about the chemical's impact on the environment, but those two are closely linked. And so understanding what your water hardness is and alkalinity is definitely going to impact your decision on which product you choose to use, which formulation, chelated or sulfate, and then how much you choose to use of it. For example, up in the foothills in alpine lakes, where you're going to have very low alkalinity, you might not be able to use more than 1/4 of a part per million of copper before you may start impacting some of the more sensitive fish species, such as trout, in those lakes and environments. So understanding that while these chemicals are solutions, they all have risks, and it really does require, doing a little bit of research on not just your water body, but what formulation of copper you're using. I often see folks just go on Amazon and Google, algae killing chemicals. And when I'm out in the field, I find all kinds of strange jugs and bottles and bags of products in people's sheds, some of which are highly questionable. That being said, one other component I think is really important, and Doug, if you're available, I'd love for you to chime in, or anyone else who happens to be on, if you want to put something in the chat or even unmute yourself, now's the time. Are you skipping ahead here? Oh, here we go. Questions, right. I forgot we put this slide in. The different formulations that we create, whether it's copper sulfate, whether it's a copper chelate, of which there are several different types of copper chelated formulations, the type that stay in suspension longer. Different species of algae respond differently to the different formulations, depending on whether they're planktonic, whether they're blue-green, whether they're diatoms. Not all algae formulations are going to function the same in attacking those cellular walls and those different species. I know that in Arizona, and that's why I called Doug if he's available. Here he is. Doug, would you mind chiming in? Because I know you guys use it in some pretty interesting ways. I would love to have your input here, especially when you guys talk about, even you guys use copper sulfate, I believe, just to address pH issues, right?
Yeah, a lot of our pHs, they'll typically run 9+, even 9 to 10 around here. We have a lot of reclaimed water lakes, and that's when we have those issues. But hardness too is one of our biggest problems. I've been trying to treat a lake that had a hardness of 1,000 milligrams per liter. And typically here our hard water is 2 to 250, which is very hard to begin with. So it's a challenge using copper here sometimes.
Yeah, I think the one way to tell if you haven't tested hardness is if you were to apply any copper formulation, it basically will turn kind of milky and cloudy colored. And that is that chemical reaction that Byron referred to, where basically your copper ions are binding and creating other copper carbonate compounds that basically just flock out of the water. And once you've bound a copper ion to whether it's magnesium or calcium or whatever, it's no longer an algicide. It's just going to settle right into the bottom of your canal or your pond, and it's no longer going to be effective. So I know a lot of people will apply those copper products and see it turn white and go, it's working. but it's actually a sign it's not working. So I think the other thing to consider here is, there's big cost differences between these different products, and that's also something that needs to be taken into account on, I think, I don't know if Kelly is on, Kelly Laurens from Metropolitan, she could speak to it, but, some of these really, really big reservoirs, just cost alone, it just makes sense to stick with the cheaper copper sulfate products. Whereas if you're getting a lot of precipitation because of hard water, then the amount of copper sulfate you may need to use is going to change that cost comparison. And so understanding how the environment is affecting the chemical choice and the formulation choice is going to really play a big role in understanding the cost and the environmental impacts of those formulations. So if there's any other questions, I don't see any popping up in the chat box. Byron, do you want to chime in or add anything to that? That's it.
I don't want to dilute your insight. That was really great. I appreciate you and Doug just, telling a little bit more about your experience and about how the water quality can really affect your decision of which algicide to use. So yeah.
I see a question here. Hang on. It says here, the copper from Hugh, the copper chart states that the lower dose is suggested when using chelated copper products. Can you explain how this dosing compares to using 250 to 300 parts per billion of Captain copper sulfate? Thank you. Hugh, I think the question, needs a little clarification. Captain is a brand name. It's a SePRO product. It is a copper chelate formulation. It's a ethanolamine and monoethanolamine, or triethanolamine and monoethanolamine chelated copper complex. Looking like you're saying to use about 1/4 to 1/3 of a part per million dosing. Is the explanation there. I'm not sure what copper chart you're referring to. Is that the chart on the label of Captain? Or
So he might just be asking, if you were going to use, say, 250 parts per billion of, a chelated copper, what would be the equivalent of a copper sulfate?
Oh, got it.
And that probably depends on the water chemistry, but maybe if you could just off the top of your head, if you're in a medium water, you could use this or.
Yeah. Hugh, if you want to send an email, I guess we could talk and I could try to answer that better for you in the specific example that you're thinking of using it in. Again, I think it's a hard question to answer because as Byron said, it definitely depends on the water quality, but it's also going to depend partly on the species of algae and what amount of algae is growing. Is it, you have a large bloom, a moderate bloom, a low bloom, and is it a filamentous or a planktonic algae or a diatom potentially? So those things would all affect the formulation you choose and the rate that you choose to apply. And then in salt water, you definitely do want to be looking into a chelated copper for salt water because of lots of potential to bind with an unprotected copper sulfate ion. Unmute. I don't can. Well, Hugh, send me an email here, or the speaker, and either one of us can help you answer that question.
Hugh, your audio is not connected.
And we can also table this, we'll have a Q&A for about 15 minutes in just about 10 minutes or so. So if everyone is kind of thinking about some stuff, or if more topics come into your head or more questions, experiences from the past, we could talk about those in, say, like 10 minutes or so.
Yeah, and one other last thing to just chime in on that is another big thing people do is they'll add different types of surfactants or additives to coppers to help them. I noticed there was a comment just now talking about mucilage and different coatings different algae will have. And so a lot of times you can get away with using different formulations if you use the right type of adjuvant that's going to help penetrate those cell walls or those mucilages that are protecting algae too. And there's lots of things on the market to help with that. Oils and soaps that are labeled and registered for use legally in California. As well as even some biologically active products that you can add. So cool. All right, take it away.
Good stuff. Thank you, Eli. So now that we talked about fighting eutrophication and, like a little bit more short-term solutions, let's go into some of the longer term solutions of how we can prevent it and we can stop it at the source. So I think probably the most popular that comes to everybody's mind is oxygenation or aeration, especially if you're dealing with water quality problems and eutrophication due to anaerobic conditions at your sediment water interface. So aeration is going to be just air, and it's 20% oxygen content. It's really tough to get your oxygen levels, dissolved oxygen in your bottom water if it's anoxic to begin with. It's really tough to get it over that 3 to 5. Whereas oxygenation is 100% oxygen, so you have a much greater transfer efficiency. The problem is that, with oxygenation, the problem with aeration is that it requires electricity to pump it. So you already have to pay that cost. Whereas oxygenation, you store it as liquid oxygen and it naturally comes out as gaseous oxygen through the pressure differential. So you don't need any electricity for oxygenation. So in the majority of cases where you're dealing with some significant anaerobic conditions at the sediment water interface, oxygenation is probably going to be the most cost-effective and efficacious method to oxygenate that bottom water and keep that sediment water interface oxygenated to reduce the release of some of these nutrients and water quality contaminants. And there's a lot of options. Some of those are just on the diagram on the side. Read Marc Beutel's paper on inhibition of ammonia release. It's a great paper, and it goes through some of the different ideas or the options that you have for oxygenation systems. And oxygenation is awesome. You just need to remember that it does have ongoing costs. So you're either going to have to pay for electricity or you're going to have to pay for that pure oxygen. And then, sometimes depending on how prevalent that anoxia is, they can be expensive for the initial construction of these systems. And the systems can be somewhat complex, like a Spiess cone. So moving down, another popular option is alum. So alum is really just aluminum sulfate, and it's acidic, so they buffer it with sodium aluminate so that you're not just acidifying the water as you're adding it. Alum gets used in water treatment as a coagulant. So when you add it in these water bodies, you'll usually see this immediate improvement in water quality because it will coagulate everything in the water column and pull it down to the sediment. And so the idea of using alum is that it creates a flock where it's, you're putting aluminum sulfate in purely just for the aluminum ions. So when the aluminum ions interact with water, they're going to form aluminum hydroxide. And if you have a lot of this aluminum hydroxide, it's going to bind together and form this very fluffy kind of grayish, whitish flock. And you can see that in the sediment core on the right-hand side of the screen. And so this is kind of like a jello or a jelly kind of consistency, very soft. And it's ideal in like a deeper reservoir where you can just kind of keep it there, and it's very quiescent. There's low turbulence, and it's just going to sit there and be stable and bind phosphorus as phosphorus is released from the sediment. It's not going to be good in your shallow water bodies where you're going to have enough turbulence that it will mix the bottom water. If you have boats going around and waves and wind, this can just stir up the flock and get it everywhere. So it's not going to do its job as binding phosphorus from the sediment water interface, and it's also going to be more likely to go into like colloidal forms to be uptaken into fish, zooplankton, whatever. And that's why there's a little bit of concerns with toxicity because these flocks are perfectly stable between this kind of narrow range of like 5.5 to 7.5. It's perfect. And you're going to have 99.9% aluminum hydroxide. But outside of this range, you're more likely to have either ionic aluminum if it's too acidic, or you'll have colloidal aluminum, which is if it's too basic. And these can both go into the food web. And this is especially true if it's in a soft water. So even if you're within this pH range, if you're in a soft water, you still have a high prevalence for creating some of these like ionic or colloidal aluminum, which can be toxic if they bioaccumulate. And one of the last ones is a little bit newer, so it's Phoslock, and it was developed in like the 90s by the Australian government. They use it a lot more in like Europe, where they ban aluminum because they don't actually want the sulfate added to the system. It's not really because of the aluminum, so they can use like polyaluminum chloride and alum. or in Europe, but generally they're not allowed to use alum. So they like using stuff like Phoslock. It's becoming more popular places like New Zealand or South America or China. It's pretty interesting how they came up with the idea. So clays are these layers of aluminosilicates, and they're you can see up here, they're the blue bars on that like unmodified bentonite that are sandwiching those ions. And so those aluminum silicates have a negative overall surface charge. And so they're held together by these positively charged ions. So usually it's sodium or calcium or magnesium or potassium, and they substitute lanthanum in there. And the lanthanum has an extremely high binding affinity for phosphate. So by putting it in this clay, they can send that lanthanum down to the sediment water interface where it'll stay, and it will form like a barrier on the sediment. And anytime phosphorus in the form of phosphate is released, then it will bind up with that lanthanum. And it forms this mineral that's insoluble, it's stable under a lot of different redox conditions and pHs. So you're essentially pulling the phosphorus out of this equation and permanently removing it. So I would say it's kind of like alum 2.0. It's definitely more expensive. So, you know, in a situation where alum could work well, like in a super deep reservoir with the perfect pH and you know, medium hardness to the water column, you know, it might just not be cost effective. But it does have a lot more efficacy as far as like when you add it, it's a clay with an ion in it. So it's not really going to impact the pH. And it's going to work in a lot wider range of pHs from like 4 to 9. So you don't need to worry about if your sediment naturally transitions outside of the range of that, you know, 5.5 to 7.5 for alum. And it's okay in softer waters. It's not ideal, but it's much better than something like alum. It's not going to be good in very, very soft water. So in something like that, I would say alum and Phoslock are kind of out of context, and you should be using something like a lime where the calcium can bind a little bit of phosphorus, but you're also improving the you're making the water a little bit harder. It does have some coagulant properties, but it's certainly not as good as alum. So it's more of a long-term help for the system rather than seeing an immediate wow factor of immediate improved clarity. It's like over the period of months and years that this works. But it can enhance the sediment stability. So when you're adding this clay, especially with like a cation in it, it's your sediment usually has a negative charge. So these cations can help to bind them all together and kind of compress this muck down. And then the clay is very fine particles, so it'll fill in all those little gaps. Another cool thing is you can cover dormant cyanobacteria. So a lot of cyanobacteria, when the light and nutrient conditions are getting low in the fall as it's transitioning into winter, they'll produce these dormant cells, and they'll go and sink and chill in the sediment, and they'll pop out whenever the light and the nutrients are back. And so this gives them another advantage to outcompete some of the other, you know, like green algae or diatoms. Well, with Phoslock, you can cover these up, and you know, you're covering them with clay, so they're not going to see the light, and then you're also restricting them from their phosphate nutrients. So they might see some nitrogen, but they're not going to see very usable phosphorus. And then the final method to stop eutrophication would really just to say, okay, you know, none of these options are really going to work for us. We just need to start over at square one. And you know, dredging is really just giving up with the system that you have and saying, okay, this sediment is just too much to deal with. We can't handle it anymore. We've got to remove it. It's extremely expensive. You got to figure out where you want to take that contaminated sediment. So it could be a great fertilizer if it's just contaminated with organic matter and nutrients. But just this whole process is extremely expensive. So if you can get away with a different option, then that's the best. And definitely don't go out dredging if you haven't secured your watershed and you haven't limited external loading. If you're still dumping nutrients into your water body and you're dredging, I mean, you're running around with your circle. Like it's a bad idea, because you're just going to have a eutrophic lake in another 5 years. And
Real quick, to kind of go back to what you were saying, there was a couple interesting questions that popped up, but I wanted to build on what you were saying about Phoslock. The one thing, what he just said about, you know, managing your watershed also applies for alum and Phoslock and other sequestering agents. You know, it's going to help. But if you're not eliminating the sources, you will end up needing to do this annually or every couple of years in order to try to address that. So sometimes you can't address those watershed issues, and this is your only solution. But another big benefit, which I don't know that was talked about enough, is really emphasizing, and there was a question about it in here, really emphasizing how alum can really affect the pH of your water and then ultimately harm your fish. And that's a big benefit to the Foslock. And while there are resources available to try to look into that, nothing really can supplement actually getting 6 beakers of your source water and figuring out different alum dosages and really testing and seeing which dosages are gonna affect your pH the most. And there are different types of alum that some are buffered and some might be a little safer to use. But I recommend it's really best to contact a company like SePRO for assistance with using Foslock, or the manufacturer of the different alums. A lot of times those guys will come out with several different types of alum and do tests with you in the field on little water samples. But it can be pretty dangerous to go out there with some bags of alum and start tossing it out there. So be cautious with that approach.
Yeah, that's a great point, Eli, is that, yeah, with alum, you're definitely going to want to do some jar testing and to figure out, what dose you need, how it's going to react. And like you said, water quality and, the hardness, the pH, its buffering capacity, all of these things are going to have a huge impact on, if you can just put straight aluminum sulfate, or if you're going to have to buffer it a ton. A softer, higher pH water, that it's going to react like crazy with aluminum sulfate, whereas something that has a lot more carbonate in it, it might be able to buffer that addition of alum. But it also, you're going to need a lot of just help as far as figuring out what dose to use. You don't want to just over drench your system and put way too much in there. And you certainly don't want to under apply it, because then you're not taking care of the problem.
Have you ever heard of any of these flocculants floating? Somebody asked about flocculants floating. I've not experienced that myself.
I haven't either. Alum
Right.
should become dissolved, and Foslock, the clay will eventually settle. I think if you just throw some Foslock on top of the water body, then you could have some air pockets in that clay. But usually, what's a better way to apply Foslock would be to vortex it and to get it into like a slurry solution, and then to apply that. So yeah, if you could see on this picture, they're injecting this slurry of Foslock rather than just kind of throwing the clay out there.
Same thing with the alum. I know some people do just throw bags of alum, but to slurry the alum is if nothing else, you're gonna also ensure you're getting better coverage, more uniform coverage with the product, whichever product you choose.
And so just finally, a last slide is just, what is the future outlook of eutrophication? So with climate change, we're looking forward to higher temperatures and a longer warm period in a lot of places. That's good. That's going to grow more algae and give more preference to an extended period of algae growth. We're also going to have more carbon dioxide, so more photosynthesis going on. I put some question marks behind urbanization, because with this pandemic, it's not so clear that people are just going to continue moving into cities, which could be a benefit to some of the more urban reservoirs and lakes if people stop forming these massive massive communities with 10 million plus people, because there's just a lot of nutrient rich runoff in these cities and these urban environments. With population growth, we're going to need more food to feed more people. That's going to require more farming and the use of more nutrients, and that's almost inevitably going to result in more nutrients going into our water bodies. But I think what hopefully exceeds all of these is that we're going to have more experience, more knowledge, and we're going to have a better understanding of how to manage our water bodies and how to effectively combat the the impacts of eutrophication. So I'll just leave with the quote that knowledge is power, but enthusiasm pulls the switch. And so thank you everyone for the enthusiasm that you bring to the lake management community and for your service to help protect waterways. So here's a little summary of everything I said in case you went to sleep or had to use the bathroom or whatever. I welcome any questions and we can have hopefully like a cool discussion right now. We got another 10, 15 minutes.
Will this be on the test?
Some of it will, yeah. Some of it will be on the trivia. So definitely pay attention. I just wanted to field some of the questions that we got in the chat over your talk, Byron. So the first one was from Emmanuel, and it said, talking about large lakes and reservoirs, are there validated successful case studies for floating wetlands? And I believe that Ellen kind of followed up on that lower down. So if either of you could speak to that, that'd be cool.
I will let Ellen speak to that. I'm definitely not an expert on the floating wetlands.
Oh, I'm not an expert on it either. I just have a colleague up in the Klamath Falls region that just put together a lit review. They were considering, U.S. Fish and Wildlife Service was considering using them in Klamath Lake up there. And so she went through the literature, and it is a article or a publication that could be cited. So I'd be happy to share it with others as well. I just sent it directly to Emmanuel.
Yeah, I think the most important thing to think about whenever we start talking about any of these methods is that not any one single thing is going to be a solution, and not any one single thing is going to be a one-time fix. So if you choose islands or if you choose Foslock or aeration, it's not that you can just choose one. Often we have to try to use all of them. So nothing is going to be a silver bullet in these environments, and these environments are going to be constantly changing. So trying to start thinking about all of these solutions as all little pieces of a whole, and the more things your budget allow you to do, the more methods you can remove nitrogen, remove phosphorus, change the redox scenarios in the bottom of lakes. The more of these solutions you can implement, the better results, sustainable results you're going to have long-term in a lake. I know we often kind of just want to hear like, what's the fix? What's the solution? And it really is trying to use all of the tools in your toolbox. And that's what we talk about when we talk about that integrated pest management approach. That you're going to have success if you can try to implement a lot of these strategies.
Thanks for that. I had a question also. It was about peroxide application, and peroxide is a strong oxidizer. And I was wondering if you could think of any negative side effects of adding a strong oxidizer to your lake.
I think there could potentially be if you just really overdose it, but peroxide is like a very quick reacting oxidizer. So generally under the labels for whatever peroxide-based algicides, it's very, very limited. So, I mean, that's the other thing too about it's much more selective in its toxicity for blue-green algae, because the amount that it gets diluted when it hits the water body, I mean, you can have fish a foot or two away from it, and it's going to react with all of the water or in the vicinity. And so it's not actually going to be harmful to the fish. But I think that also works against it. So peroxide is very, very safe to use, but it's just because it's so safe, it's going to take a ton of using it, and you're going to need to spot treat all of the areas with the algae, and it's not going to really persist for more than a couple minutes. It also makes it sometimes difficult too, when you're trying to use the solution that's readily reacting with everything around it. So, you need to kind of be quick with using it, and just very precise in where you want it to go.
Thanks.
Hey, I had a comment about the floating islands.
Okay, cool.
Sure.
Yeah, this is AJ Jones. I had actually done this calculation before to see theoretically how much these floating islands could remove, how much phosphorus they could remove. So the limiting factor in that is the plant's biomass. So from doing a little research, I think the highest phosphorus per biomass for any aquatic plant is going to be about 3%. So if you were going to remove, let's say 100 pounds of phosphorus from a lake or reservoir, the actual biomass of the plant would have to account for about 3,300 pounds at the highest level that they can store. So it comes to a point where that's the limiting factor. So unless you can actually remove that biomass over time, then the amount that you're going to be able to remove is not going to be a substantial amount unless you have very large water floating islands.
Yeah, I think that's a really good point. But that also is just to remember that Byron had mentioned too that, you know, there's a lot of other benefits to those in terms of zooplankton shelter and some nitrification and some other factors, shading and habitat. So, but in terms of phosphorus, yeah, I've heard that before. It's not exactly the best phosphorus removal strategy.
Yeah, that's a good point.
So I don't know if we touched on this one earlier when we were talking about different algicide options, but Larry asked which algicide works best on saltwater lakes.
Yeah, honestly, it's not using coppers really to begin with, but one of the issues you're going to have with that situation using coppers in salt water is typically they probably are, at least in California, connected to the bay, to the ocean, to intertidal areas. So there's going to be some pretty important regulations that are going to probably limit your ability to use coppers effectively within the requirements of the law. So often people don't use coppers in saltwater environments. But in general, yeah, you would have to be using higher rates if you were to have effective results with coppers because of all of the binding that will occur even with the chelated version. So not typically recommended to use coppers in salt water.
So this next question, it's from Carrie Austin, and I think it's related to algae control. Can someone say more about adjusting fish populations? Are there multiple examples in California, including obtaining permits from CDFW?
Yep, that's a very great question, and something I should have brought up is that, yeah, if you're, if you don't own the lake or the reservoir yourself, then you're definitely going to need, and even if you do, you're going to need approval from California Department of Fish and Wildlife. And so there are some examples. I think the Army Corps of Engineers are generally the most successful with biomanipulation and fish stocking and removing carp for benefiting the ecosystem. And that's just because humans tend to think we're really smarter than we are. And nature is really, really intelligent, so a lot of times when we try to do something to manipulate the food web, like, okay, well, let's just get rid of the wolves, that has a dramatic impact in the long term. And so unless we're very, very calculated with our approach, it's not a good idea to just say, oh, okay, well, I saw too many prey fish in this area, so I'm just going to throw 1,000 largemouth bass in my lake, because they might eat everything, and then they might not allow for a habitat for small fish. And then that could, dramatically reduce the entirety of the food web, and then you might not have any fish to put those nutrients in. I think Alex might have some insight on this. He's worked with some cool cases where biomanipulation was a tool that they were kind of analyzing.
Can you hear me? This is Alex.
Yep.
This is Alex. We have a wonderful experiment in Mountain Lake. We took out all the fish, dredged the lake, and got a Secchi depth. We then added stickleback only, and the Secchi depth, as you might expect, went down because the zooplankton. So I think the point, Brian, that you're making here, you've made it earlier, is that it just go through the zooplankton, and they need somewhere to hide, either in the shade or the dark, as you mentioned, possibly in your floating islands even, or down at the bottom if there's oxygen, or in the weeds at the edge. And this lake, we haven't managed to establish the weeds on the edge. And then the unfortunate turn is that people ignored our notices and have thrown in largemouth bass, which have now wiped out or reduced very much the crayfish. So I'm waiting now to see what the Secchi disc will do. But I think biomanipulation has to be the ultimate goal. you know, all the other methods should lead to that, because if we get a functioning biomanipulation based on what you've given us here in the talk, we can reduce the amount of these other controls we need to use, whether it's alum or oxygen or herbicides or algicides. So I think it's unfortunate for a science point of view, but we may have to use multiple methods of lake treatment, which means it's going to be harder to pick out individually which one worked best. Unfortunate for science, but good for us.
What about using bacterial additives? Now that's something I've used a lot of, but there's limited research. Byron, what have you learned about people using Pseudomonas and Bacillus strains of bacteria and other things to compete with algae? Is that something you've looked into?
Yeah, I'm obsessed with the idea of beneficial bacteria for like nitrifying bacteria and phosphate accumulating bacteria. So they're proven concept for sure in wastewater treatment. They will grow these huge colonies that really stimulate some nitrification, denitrification, some phosphate accumulation, some of that good stuff. I'm not so convinced. It's kind of like, in my opinion, it's like probiotics for a human being. So as long as you're adding the bacteria constantly, then you can probably keep those communities established. But I'm not so sure that they can actually establish themselves in the natural environment, because you have to think that they basically have to outcompete whatever's naturally in the sediment itself. So if you have, you know, a lake that's transitioned a lot from, like, it used to be very oligotrophic and it's transitioning into eutrophic and hypereutrophic, that might be a good time to seed it with some of these beneficial bacteria that can kind of do this nitrification and denitrification and phosphate accumulation. But if you've had a eutrophic lake for a long time, then the bacteria in there are probably already, you know, they're the best at doing these things, and they're the most competitive for getting at the ammonia and the nitrate and the phosphate. And so I think sometimes seeding this bacteria, it's almost like someone taking a probiotic for a week and thinking that that's really going to change your microbiome. Like the evidence doesn't really suggest that, but it does suggest that when you're consistently taking a probiotic, your microbiome will be different while you're constantly inputting those bacteria. So I think it's complicated, but I have a lot of hope for it. And
so regular dosing will keep your lake regular. Is what you're saying.
Yeah, man, if you want to come out with a brand of beneficial bacteria, that's a perfect slogan. And I have heard some interesting things. I don't know if there's you know, scientific evidence and journal articles about this, but I have heard about beneficial archaea. And so there's some interesting archaea that are really good at you know, nitrogen cycling and uptaking a lot of phosphorus. So I felt like that was interesting stuff, but this is also on the, we don't have a ton of evidence to suggest that this will really fix your lake, and you know, it's at your own risk.
Yeah. Again, I would say, you know, keeping that in, you know, when I hear about people using it and people requesting to use it, I always make it clear, you know, it can be used, but it's part of a bigger program, as Alex was just saying too. There isn't going to be the one solution, and a lot of times we do have to rely on lots of these.
And another thing I'll say about the beneficial bacteria, which I find really interesting, is that I have come across some strains that seem to be good at like oxidizing organic matter and reducing muck. And so that could be a huge advantage. But the side effect of that is, it's just like changing a microbiome from a bad one to a good one. You're going to have some side effects along that transition into a healthier environment. Is that when you're oxidizing all of this organic matter in the sediment, you're going to be releasing a lot of these nutrients. So I have heard anecdotal evidence that, you know, they applied this and there was these blooms, you know, and algae blooms afterwards, which I would say is a good thing, because you're getting rid of all the nutrients that you're storing in the sediment to get rid of later. So that's just something to keep in mind, is that you might put something in there that would help degrade your muck and the built up organic matter in the sediment. And in turn, you might have some short-term algae problems.
And that's Alex, also known as Marcy, made the comment too that you really do. If you combine the discussion of bacteria with the with the redox model he showed, the redox ladder, if you don't have aeration and oxygenation, then you're going to have some limited results if you with any of that benthic community at all. So
Byron, here's another question from the chat from Patrick at Turner Designs. Are the algicides that are being talked about deployed when algae blooms are in full swing, or are they deployed pre-algae bloom?
Yeah. So the answer to that is going to be a little bit of both. You can have kind of preventive algae dosing, copper algicide dosing, because copper is a little more, does stay in the water for longer. It has a longer residence time. So you can do low dose treatments ahead of time. You can, to some degree, predict some algae blooms by looking at certain water quality parameters. And even lake managers, just purely based on temperatures, will know you've got a heat wave coming, and they will come in with low dosages to kind of have a preventative effect on algae growth. The one thing and then typically people do use them reactively to control algae blooms. That's one thing that's really important, which we didn't discuss. And Byron, if you want to address it, is dropping of DO, loss of DO as a result of killing algae. You want to take that one on?
Yeah, I would say that that's my biggest hesitation on using algicides. So looking at the research, there's some good research that shows that copper doesn't really accumulate at a high rate, even if you're consistently using algicides. So that's kind of that hesitation about the use is overblown, in my opinion. Like, if you look into the science, you're not applying enough copper usually to impact the sediment, which already has a naturally somewhat high abundance. And this copper is getting bound up and getting bound with organic matter. It's getting reduced into the elemental form a lot of times. So that's not the issue. But the real issue that you have to worry about is it's just a short-term solution. So it's just like in that animation, you saw all the dead algae goes to the sediment water interface and results in the reduction of redox and the depletion of all this the oxygen. So you've got to think about that. And that's why I think, the PCAs, they always talk about, if you have a huge algae bloom, you can't just treat everything at once. You have to treat in small sections so that you're not depleting all of the DO in the water column, because it's not actually like the algae that's going to kill fish. A lot of times these fish kills are simply because someone treated all of the algae on the surface, which covered the entire lake, and it was a ton of it. And when all of that dies at the exact same time, now the sediment is just going, the bacteria in the sediment is just going to explode and use all of that organic matter and deplete all of the oxygen. Even sometimes in a shallow lake, it's not fast enough to mix oxygen through the atmosphere into that water body as those bacteria are going so crazy, eating everything up. And so that's what we kind of got to look out for. And that's why I think that, if you're going to use an algicide, we need to be looking into, what's happening with these nutrients, because as these algae die, their nutrients are only getting stored in the sediment for a set period of time. They're eventually going to be released.
Great. Well, I think we're coming up to the end. And I'll just say, Byron, you're going to be on the expert panel on Friday, right?
Yeah, I don't know if I qualify as an expert though, but
You do now.
Talk about
W expert. Alex agreed with you on more than one occasion. That makes you an expert. In fact, I don't think he disagreed with you once, which is like a miracle.
I think we need Alex on. I'm gonna have an earpiece so that he's.
Oh, he's speaking to you on his cell phone too at the same time.
It'll sound like I'm the expert, but it'll really be Alex.
Yeah, Alex is our founding father. So I will say I see we did just get one final question. Patrick Turner Design, one of our sponsors, and I also use their fluorescent devices. I have one of their handheld devices, and there are now lots of devices we can use out there which are pretty new overall in terms of monitoring and starting to get real-time data without having to send samples into a lab and wait for results. So while some people may make arguments about the accuracy of those, I think that that's somewhat of an irrelevant argument, because it does provide a trend in and of itself. So having fluorescence monitoring, you know, relative to itself will show trends in lakes, whether it's deployed in real time and reporting over an antenna, or if you just have someone out there collecting information daily or different times. Those are really great tools, really great handheld tools that you can have in the field. So I recommend those for sure. Mark, do you want to do the final wrap up? And guys, I see there are some other questions, but we're right here at the end, the last minute. Any final talks?
Yeah, we have tons of questions. Byron, would you mind sharing your email in the chat so people can follow up with you directly?
Yeah, sure thing.
Thanks for that. Cool. So yeah, thanks everyone for joining. We're going to have talks for the rest of the week, and along with our lake trivia or expert panel I was talking about earlier. If you want to check out the schedule, you can just go on calms2020.org and look at the whole schedule for the week. Ask your friends to register. Stock up on beer for trivia. And yeah, with that, yeah, thanks everyone for joining. Thanks Byron and Eli for a good talk.
Yeah, it was my pleasure. Thanks you guys. Good job today. You killed it.
Thank you.
Thank you.
Thanks, everyone.
All right, that's it, man. Good job, buddy. You killed it, man. That was awesome. I even took some screenshots. I'm gonna steal some of your slides, man.
Yeah, that was a good talk. Thanks.
It was cool to have both of you guys in there and, you know, have a cool discussion at the end. I like that.
Sure.
Yeah, there was a lot of really good questions that came up. I was